So 2.6 is all about the structure of two nucleic acids, namely DNA and RNA. And DNA stands for deoxyribose nucleic acid, and RNA stands for ribose nucleic acid. So they're both nucleic acids, and nucleic acids are polymers, which are made up of individual monomers called nucleotides, which we'll look at the structure of in just a moment. The essential idea here is that the structure of DNA is all about storage of information, storage of genetic information. And one gene is a length of a molecule of DNA. And here we can see the chromosome. The chromosome is the structure. And chromosomes are found in the nucleus of eukaryotic cells. But if we unravel a chromosome, it's made up of this material here called DNA. And we're going to look at the structure of this in detail. But what you can see, these letters here are called bases, and we've got a length of DNA has got what's referred to as a length of complementary base pairs. And a length of complementary base pairs is a code to make a protein, or strictly speaking, it's one gene, one polypeptide. But we need to think about the importance of proteins in living things. All of our tissues of our body, our bone, our skin, our muscle tissue, all of that is made up of protein. So we've got protein as the structural components of our bodies. But then in terms of the biochemistry of our bodies, our metabolic processes, we've got proteins which control that too. So we've got enzymes which are controlling the rate of chemical reactions. We've also got hormones, which are chemical messengers, which are vital for metabolic functions. We've got neurotransmitters, which is another protein which allows nerves to communicate with one another. So proteins are really, really important in the structure and the function of living things. And so we've got all of the code to make a protein, which is here in our DNA. RNA is a molecule which is also important for protein synthesis, which we're going to look at actually in a later unit. Can you go into the Google Classroom and have a read of all of this text here? Because it's very interesting, and it signposts you really how massive the DNA molecule is in terms of its structure within the human body. We've got the syllabus statements here. This quite short, but I want to jump straight into this idea of the first syllabus statement that nucleic acid as a polymer is made up of individual units, monomers, which are called nucleotides. So what you can see here is a nucleotide, and the nucleotide structure has got three components to it. We've got a phosphate part to it, and notice the negative charge on the phosphate here. This is a very reactive molecule, which is the phosphate. And here we've got a 5-carbon sugar. This is deoxyribose sugar. And up here we've got a base, and there are 4 different sorts of bases, which we'll look at in just a second. Now these numbers 1, 2, etc. are referring to the carbons. So by convention, carbon number 1 is here, carbon number 2, carbon number 3, 4, carbon number 5 projects outwards, and then we've got this covalent bond to the phosphate group here. Carbon number 1, we've got a covalent bond to a base structure here. So the nucleotide has got 3 parts to it, but the nucleotide can differ between DNA and RNA in a number of features, one of which is this sugar. So here we've got deoxyribose sugar, which is making a nucleotide, which is going to be DNA eventually. If this had an oxygen group here, then we've got ribose, which is another pentose or 5-carbon sugar that will make up what will eventually become RNA. So this slide is unpacking this a little bit more for us. So we've got the 5-carbon sugar here. This 5-carbon sugar can either be ribose or deoxyribose. The phosphate group is always the same. It's referred to as being acidic because it will donate a hydrogen and it's negatively charged. And here what we've got are the nitrogenous bases. And the nitrogenous bases for DNA are adenine, thymine, guanine, and cytosine. Now in RNA, what we've got is adenine, guanine, and cytosine, but not thymine. Instead of thymine, we're going to replace it with another base, which is called uracil. So if we look at the differences between DNA and RNA at the nucleotide structure, we've got to think about the differences in the sugar part, ribose versus deoxyribose, and also the base parts, specifically thymine versus uracil for DNA and RNA respectively. Again, can we just notice that we've got a covalent bond between the carbon number 1 here and the carbon number 5 and the phosphate just here. So DNA versus RNA, we've got ribose sugar or deoxyribose sugar. I would recommend that you learn to draw these. Just you need to be able to recognize them if you remember from earlier on in Unit 2. And this table is just highlighting those differences that we've talked about so far. The bases, the differences here, we've got uracil in RNA compared to thymine in DNA. We've got ribose sugar versus deoxyribose sugar. So the difference in the base and the difference in the sugar is at the nucleotide level. If we then think about putting those nucleotides together to make the nucleic acid, the macro structure, if you like, we get strands of nucleotides. And what we need to appreciate is that RNA is single-stranded, whereas DNA is double-stranded. And it's double-stranded and is called an anti-parallel double strand, which then folds up into a double helix. And hopefully this will be clear by the end of this little video presentation here. So we've got differences at the nucleotide level, and then we've got differences at the macro level for the whole of the nucleic acid. Now, probably what's less familiar is this idea of a base. So let's unpack that with a little bit more detail. So we can classify bases into two sorts, and they're very, very rich in nitrogen. We've got bases which are referred to as pyrimidines, and we've got bases which are called purines. And this structure here is just identifying the fact that the purine is a larger structure. If we then subclassify further, what we find are we've got two different sorts of pyrimidines. We've got cytosine and we've got thymine. And the reason I've just bolded up the Y's is that they're both pyrimidines, and all of them have got a Y within these words. So pyrimidine has got a Y, cytosine has got a Y, and thymine's got a Y. Hopefully that might make it easier for you to remember. And the purines haven't got a Y in them. So what we can see, if we look at the next slide here, this idea that we've got always complementary base pairing. And in DNA, thymine is always going to complement with adenine. And when it does complement with adenine, it's going to form these red structures here, hydrogen bonds. Here you can see that we've got a very rich source of nitrogen here. So we've got the thymine always binds with adenine, and similarly, we've got cytosine always binding with guanine. And what I want to draw your attention to is the fact that the distance between this molecule here and this molecule here is exactly the same. And that becomes important later on, and that's why we have this particular complementary base structure. Another thing to signpost is the fact that there are 3 hydrogen bonds which exist between cytosine and guanine, and only 2 which exist between thymine and adenine. So if we go back and think about this nucleotide structure, hopefully this is clear now. So this is going to be either deoxyribose or ribose, depending if it's DNA or RNA. The base part is going to be adenine, thymine, cytosine, and guanine if we're DNA. And instead of thymine, we've got uracil if it's RNA. But what we need to think about now is how do we put these individual nucleotides together to form that macro strand? So here you can see one nucleotide and a second nucleotide. And what is going to happen is that the hydroxyl group on carbon number 3 is going to react with this phosphate group, which is on carbon number 5. It's going to be a condensation reaction, so we're going to kick out a molecule of water, and we're going to produce this covalent bond, which is specifically called a phosphodiester bond, very strong bond between this nucleotide and this nucleotide. So we've got a dimer molecule that's being formed here. And if we do this repeatedly, what we're going to end up with is this kind of structure here. We're going to have a phosphate sugar, phosphate sugar, phosphate sugar backbone, individual nucleotides being held together by the phosphodiester bond, this covalent bond, and then horizontally we've got these bases projecting outwards. And if we go back to this structure here, you can see a little bit more detail. You don't need to be able to draw the individual molecules out like this. I would recommend that you know how to draw and distinguish between, is the important thing, deoxyribose and ribose. But what you can see are the bases here are rich in nitrogen. But let's go back to this one here. So we've got a single strand here. Remember that carbon number 5 is found at this end. So this part of the chain is referred to as the 5' end. And if we go down the bottom of this strand, we've got carbon number 3 just here, and this is referred to as the 3' end of the molecule. And that's going to become important when we take two different strands and we put them together. And when we put the two together, what's going to happen is this. So we've got the 5' end coming down to the 3' end, and then we take a second molecule and we turn it upside down so that the bases can face one another. And what we've got here is the 3' end and the 5'. We've got 5' and 3' are opposite. So we call this anti-parallel because we've got 5' to 3' versus 3' on this side to 5' here. Parallel because the distance between these molecules are exactly the same all the way through the molecule, and the distance is maintained because we've got complementary base pairing. We've got adenine, thymine, guanine, and cytosine always being paired up. Hydrogen bonds are going to exist between the individual nitrogenous bases when they form that complementary base pairing. Remember, individual hydrogen bonds are weak, but because we've got thousands of these, if we go down a molecule of DNA, the overall structure is very, very stable. Then what happens is this is coiled up into a double helix, which is the final structure. And it coils up into a double helix so that it's compacted and it can be easily stored. So this diagram here, you do need for one of the syllabus statements to be able to draw and label a molecule of DNA. So this is the kind of thing that you need to go away and practice drawing. It's not straightforward. One thing I normally do is I can draw this side of the molecule quite easily, and then I turn the piece of paper upside down and I draw it the other way around. But I actually literally turn the piece of paper upwards. Whenever you're asked to draw in IB, it means draw and label. Excuse me. So can you have a legend that says A stands for adenine, T stands for thymine, label your hydrogen bonds, having box sections like this one to say that this is a base pair and this is a nucleotide is always also very, very worthwhile to do. So you do need to be able to draw and label this molecule. This slide is just referring back to the earlier one. Please have a look at that and make sure it's clear. And this one is showing you again how it coils up in order to be stored. In our Google Classroom, you can have a look at this one. It's useful to review. This slide here is just referring you to the fact that you've got to be able to draw and label. And then we come to this slide here, and this is just asking you to remember that the structure of DNA can often be represented as a model. So this is actually a three-dimensional model that we're going to come to look at when we go back into school. And this is also a model. And the elucidation of DNA by Watson and Crick was done by them physically making models and a bit of trial and error. And I'm going to ask somebody to present on these last two slides here, which is looking at the experiments that Crick and Watson did in terms of the elucidation of the structure of DNA. But hopefully that's helpful to introduce you to what DNA and RNA molecules look like. Can you read the textbook? Can you look at Cognity? Also, there's lots of resources that I've posted on the Google Classroom, and then work your way through the booklet for 2.6 and we can troubleshoot next lesson. Thank you very much.